Hello everybody, good afternoon. Just left my meeting with 8 seconds to spare on the countdown, so just going to gather my thoughts quickly, and I'm going to fill slightly while I do that. So hi, how is everybody? Everybody good? It's Wednesday for the live streamers, and it's a lovely day in Newcastle. The sun is shining, it's very warm. And we've reached another divisible by 10 episode of Lockdown Learning, so number 40 today. What we're going to do today is to very much pick up from where we left off yesterday. So what we've been doing the last couple of days is to kind of recap the analysis that we were doing last week in respect of analysing our RNA-Seq data, which we did comprehensively for the NHBE cells last week, and then we've been repeating this week for the A549 cells. And the reason for doing that particularly this week is so that we have then two sets of differentially expressed genes, two sets of analysis that we can compare to each other, that we can say, okay, well, what are the similarities and the differences between these analyses? So what I would never expect going into this up front is that we would find the same genes being differentially expressed in these two cell lines. I think that would be unreasonable to expect that to be the case, that the same genes that are differentially expressed in the NHBEs will also be differentially expressed in the A549s. I think even if you did, even if these two experiments were technical replicates of one another, or in the same cell lines, you still wouldn't necessarily expect to find exactly the same genes being differentially expressed. What I would expect to see is some similarities between the gene expression in the two cell lines. So I'd expect to see some trends in differential expression being maintained between the two different types of cell. And also that the same kind of processes are being affected. So if we take a look at our differentially expressed genes and have a look at the functions, the biological processes, the molecular functions, certainly the biological processes that they're involved in, I would expect to observe some similarities there. Now, the fly in that ointment slightly is that we have quite a small number of differentially expressed genes in the A549 samples. So consequently, I would expect there to be fewer sort of biological processes represented when we look at that Go analysis, which is what we're going to do this afternoon. Because you've got fewer genes to work with, so finding enriched, overrepresented processes within that small number of genes is much more difficult to do, statistically speaking. So that's a little bit of a fly in the ointment, and we can maybe think about ways in which we can approach that as we go along this afternoon. So what we want to do first of all is get ourselves to the point with this second analysis that we got to with the first analysis. And so we had got to the stage yesterday where we had got we'd looked at the visualizations of our differentially expressed genes, and we had retrieved our annotation as well, so that we could produce our interactive volcano plot with gene names on. The next stage then after that is then we did a heat map of the differentially expressed genes, so we can do that again first up, and then we did the pathway analysis. So they're kind of our two jobs for this afternoon. If we get sufficiently through those jobs, then maybe we can start to think about looking at the comparisons between our two sets of data. But that may be reserved for tomorrow, depending on how far through we get. Okay, so I'm gonna turn my front page off. There we go, and there we've got our R session. I have restarted my RStudio since we last spoke, because I've rebooted my machine, so I'm just checking that the state is as it was when last we spoke. So I've got all of my data loaded in. So I saved my R session and reloaded it. So we're in a good place. So the first job then, therefore, was our heat map of differential expression. So we produced our annotated volcano plot. That was the last thing that we did. Yes, so that's our volcano plot. I'm not going to make it interactive because it slows everything down horrendously, but that's our volcano plot there. So the next job then is the differentially expressed genes, for which we want our annotated data frame. So we want to filter our annotated data frame down to our differentially expressed genes, because we haven't done that yet. So we've got A459 ANODF. That is effectively our A459 filter 1, but with annotation. And so we can filter that down further to get us to our third filtered, so thresholded on p-value and fold change dataset with the annotation. So these are the filters that we applied. We just have to apply them to the correct dataset. So we'll filter that one to produce A549. We're going to call it ANO2 now. And then we'll do the second one, the second filter on that product of the first filter there, in order to get us down to just our differentially expressed genes. NA is introduced by coercion. What happened there? Okay, that's interesting. I know why. Because I've restarted, but I haven't reloaded my packages. That's what's happened there. So that was using the base filter function, not the dplyr filter function, and consequently got very confused. And then so that's our first filter there, 279 genes, and then the second filter there gives us, it was 60 something, 59 genes. So yes, that's our differentially expressed genes with the annotation, and that's important for giving us the row names that we want for our heat map. That's why we want the annotation in that data frame. Okay, so data for our heat map. We already have this this matrix of VST data because we used it yesterday when we made the PCA from scratch. So yesterday we built this PCA here, and in order to do that we got the VST transformed data as a matrix. So we already have that data ready to go, so we don't need to generate that this time because we've already done it. So that's one step that we can save. But then we need to filter that down to our differentially expressed genes. Okay, so the differentially expressed genes are the ensemble gene IDs from here. So let's make sure that we keep our variable names different. So A549 DEGs there. So that should be a vector of ensemble gene IDs. 59 ensemble gene IDs. There you go. There are differentially expressed genes, and then we can use that to pull the appropriate rows out of our matrix of variance stabilizing transformed data, like this. And then we add the gene names, and then we can produce the heat map. So let's do that. Okay, so let's call again, sticking in different variable names as we go to make sure that we're not overwriting the same variables from between our two different data sets. I've forgotten the name of the variable for the VST matrix. Let's have a look in the environment. It is VST underscore VST underscore mat, sorry. VST underscore mat. And it's A549 DEGs. Make sure we get that right as well. Otherwise, we end up with differentially expressed genes from the other data sets. So that's that will be the matrix of the expression of our differentially expressed genes after a variance stabilizing transformation. And then we want to set the row names for that matrix to be the external gene names. So that's the human readable external gene names. So let's run these little steps to give us a heat map. A matrix of data that we can put into a heat map with gene names as the row names. And then because we don't have that data, it's called A549 underscore HM, not data for HM. There you go. That is the heat map produced by base heat map of our 59 differentially expressed genes. Okay, so if you remember the things about the heat map, the clustering across the columns. So we've got our one set of our samples clustered into a group of 3, and these will be the mocks because the expression is low. No, the expression is higher in these, so they should be the infected samples. And then we've got on the other side, we've got our the other samples with, in this case, looks like lower expression. And then we've got clustering across the rows as well. So the rows have been clustered into genes which exhibit similar patterns of expression. And it is patterns of expression because this is scaled by row. So it's not that they exhibit similar expression across the data set. It's that their patterns of expression are similar because everything is scaled, is mean scaled across the rows. So you've got a scale done by subtracting the mean and dividing by the standard deviation to give you this z-score across the rows. Although none of that is documented in the legend of the heat map, it's not shown how that actually works. So instead of using this base heat map function, we instead relied on the pheatmap function from the pheatmap package, and and we use that instead to heat map our data, which we ended up quite liking. So again, this is our heat map, this time for our 59 differentially expressed genes. So there we go, that's our heat map of those differentially expressed genes, and looks quite nice. You can see that we've got, these are our, there's clearly upregulation here in these samples, so the expression here is higher in the samples on the left and lower in the samples on the right, and there's a lot more of those genes than there is the block at the bottom where the expression is lower on the left and higher on the right. So these are the upregulated genes and these are the downregulated genes, and we've got on the left we have our infected samples, and on the right we have our mocks. And I think that's it's easier, it is immediately easier to interpret this heat map, because we know what colour represents relatively what expression, whether it's higher or lower expression. We know from the colours what's represented here. So that's worked quite nicely for us. It's a nice little heat map. And so I'm not going to play with the colours like we did before, because actually in the end, although I'm not 100% sold on the diverging colour scale of this data, because it's kind of linear expression, but that normalization within row normalization gives you a data which has a center. So the diverging color scale makes some sort of sense, at least in this regard. So although I'm not 100% sold on it, but I think it's okay. And I think it's a darn sight better than color schemes I can come up with on my own, because I'm just not very good at colors. And if any of you have a better design sense than I do, then maybe you can do a better job of this than I can. So then the final thing that we did in the original analysis was the Go enrichment. So this was looking at the gene ontology and finding overrepresented gene ontology terms amongst our differentially expressed genes. And as I've already said this afternoon, I am slightly pessimistic about the potential for success here with this particular set of genes, just because it's so small, because 59 genes is really, really not very many in the grand scheme of things. And I don't hold out a huge amount of hope for finding a large number of biological processes being enriched in this data, purely because it's such a small number of genes. But we may as well suck it and see. Let's see what we get. And hopefully if we do find anything, hopefully there'll be similar terms to the terms that we had before. So, okay, to run this, I'm just going to copy and paste this code and then edit it in place, and again changing the variable names as we go, so that we're not reusing the variable names from the first analysis. And the reason for that, as much as possible, I've tried to change all of the variable names in this second script. I may have missed a couple, but I've tried to change all of them. And the reason for that is that at some point I am going to want all of this data together in a single environment. In one, I want to load them all into the same R session, so that we can compare the first analysis with the second analysis. And it's going to be much easier to do that if the two analyses are already using distinctive variable names. If the difference between the first data set and the second data set is obvious in their variable names. If I just copy and pasted this code and just changed the sample filtering at the very top, so just kept all of the same variable names, but just changed this filtering here, then I wouldn't have been able to read both in simultaneously into R without then going through and changing all of the variable names in at least one of them. Because if I'd have reused all the same variable names, then when I read the first analysis in and then read the second analysis in, the second analysis would completely overwrite the first analysis. And I would not then be able to compare the first analysis with the second analysis. So I have tried, where possible, to be quite diligent about changing those variable names as I've gone along, so that I'm not reusing the same variable names except where data is shared between the two analyses. And there's very few places where that data is absolutely shared. So the overall sample table is shared between the two, and the gene map is shared between the two. But actually, other than that, almost every single piece of data that works its way through our analysis here is distinct between the two analyses. And so I hope that I've kind of caught most of these things, and then we're not going to end up without things to compare between the two once we run both scripts. So with that in mind, let's get renaming variables here as well. So this is the NHBE gene ID. So the first thing that we do for the Go analysis is we need these particular form of identifiers for the package that we use, the cluster profiler package that we use to look up the gene ontology annotation in the database that it uses. So although we found that cluster profiler doesn't necessarily rely on NHBE gene IDs in everything that it does, it relies on them enough that they're the identifiers that we want to use for this analysis. So we want to retrieve them from the right data set. So that is our A549 ANO 3 data frame. That's our differentially expressed genes. We also had to turn that into a vector. So that's what this line does, is it turns that data frame that's returned by getBM into a vector, and then we want to ensure that that vector is a vector of characters, not a vector of integers, which is what it comes back as, because the integers don't work for everything that Cluster Profiler wants to do. And then we want to do the same for the universe, so the gene universe, which is all of the genes that we analysed for differential expression in DESeq. So that's everything, all of the genes that passed a particular DESeq, the particular DESeq filters. So that's the total count greater than 0, the base mean greater than the low counts threshold, and the Cook's distance threshold as well. So all of those filters are applied before we do any testing for differential expressions. So we don't want to consider those part of our universe if they've been filtered out by DESeq. So that's this A549 ANO-DF is the data frame that contains just those genes which are tested by DESeq, around 16,000 or so genes. And again, we want to set that we want to get the results from Biomart and then make it into a vector. That's what. Second, that line does, gets it as a vector. Let's get rid of the as character there, and then that line turns it into a character vector there, just making sure we change all of the variable names in all of the right places. Right, so then we're going to run the Go enrichment calculation over the biological process ontology. So our list of differentially expressed genes is this NHBE A549 vector, and our universe is the A549 universe vector. So those are the things that get plugged into our enrichgo calculation, and we better change the name of this as well, trying to be nothing if not consistent. Okay, so these few lines of code now will extract the appropriate data and then run the gene ontology enrichment calculation over that data to give us the enriched gene ontology categories in the biological process ontology, and then we can use the summary function with our enrichgo results to see what they look like, to see what those results look like, see if we get any results with our 59 differentially expressed genes. Okay, so summary ego A549. And I'm going to view this. It does at least look like there's some results, which is good. I was worried there wouldn't be any at all, but it does look like there's some results. If we run view with that summary, then we can see what results we're getting. And we're seeing, okay, so vaguely similar. It's a bit more viral. The other cell lines seem to be a bit more response to bacteria, response to kind of inflammatory response, whereas this is response to virus, interferon response, interferon signaling. So very much, again, looking at response of cells to pathogens and an inflammatory response going on here. So yeah, I think a lot of what you would expect, and it looks, relatively speaking, looks similar-ish to the case for the other for the other experiment as well. There's some similarities between the responses here, which is good, nice to see. And so again, I couldn't get some of these visualizations to work with the last data set, so I don't know if this is going to work this time, but we can have a look and we can see. So you've got the bar plot, which for some reason, it's being truncated because of the length of these labels, so it needs more space to try and draw. So I'm just going to zoom my plot to give it a bit more space. There you go. So it needs a bit more width because the names of the categories are very long. It needs a bit more width to be able to actually plot the bars properly. So there you go. That's the bar plot. What were the other ones? So the dot plot will be similar, and will similarly suffer from the same sort of problem. Ego 549. I've got to plot the right thing. There we go. So again, we can't see the dots unless we make the plots pane take up the whole window. In which case, the labels will fit, and we can actually see the plot, which is fine. So this top thing here, more gene defense response to other organism, more than half of the genes that are annotated with that term in the whole gene ontology, in the whole biological process gene ontology, are being found in this gene list. So the gene ratio is greater than 0.5, so it's more than half of those genes coming out in this analysis, which is interesting in itself, I think. So then let's see if we get a CNET plot this time, see what happens. Because this didn't work for me last time, although in the comments at the time it seemed that it did work for others. It has worked for me this time, so that's nice to see. It's slightly... I'll zoom it in again just so we can talk about it a little bit, because I didn't get the chance the last time. So, wait while it redraws. And so this CNET or connectivity net plot is showing you the shared membership between the different biological process categories. So this is I've one of the arguments I passed to the plot here was show category 5, so it's only plotting the data for the top 5 enriched Go terms, but it's showing me whether there are any genes in common between those 5 sets. So it's drawing. It's included the genes on the plot, and then you've got edges between the genes, which are the grey dots, and the categories themselves, which are the kind of browny, beigey dots. There's an edge between the gene and the category if that gene is represented in that category. So this is showing you the overlap between these different categories and whether there are genes in common between them. And then the layout is trying to then show you the groups that share genes in common. So you've got this gene up here is only shared between these 2 interferon pathways, processes here. This group down the bottom is mostly shared between these 3. They look like effectively viral response terms. Then you've got some groups here which are shared between both interferon and viral response categories. So this is quite a useful visualization, I find, to customize this a bit so that the labels on the genes here are the NHBE gene IDs, which is the only information which we passed to our the calculation to find these enriched Go terms. You can make this analysis readable. That's how Cluster Profiler describes it. So let's go back to all the panes. There's an argument that you can add to your enriched Go when you run it, which is to do with readability. So readable equals true, which will map the NHBE gene IDs back to gene names. And if your analysis is readable, then this CNET plot should include those gene names on the should include the gene names on the nodes rather than the NHBE gene IDs, which indeed it does. So now you can see which genes are shared between which categories. You could also make this bigger, so you can increase the number of categories. I think you can choose to color the nodes in, so you can color the gene nodes in by their differential expression as well. Try and remember the argument for doing it. There you go, fold change, and you would have to then pass it the appropriate mappings between, I think it would be a named vector. Let's see if we can figure that out, because it's worth doing. And we've got some time, so let's do that. CNET plot. There we go. Fold change. Just says fold change. That's not very helpful, is it? I would imagine it's a named vector. Let's see what we can do. How to do this? It would be the so fold changes equals it's from our A549 Anno 3, yeah, A549 Anno 3 and it's the log2 fold change column. That's our fold changes. And then the names of that vector should be the external gene names, because that should provide a mapping between the fold changes and the genes that they represent. See if that works. Oh, that worked nicely, sort of. So they're all positive, which I guess is not massively a surprise. An awful lot of these genes are positively, do have positive fold change. So the color scheme, because they're all positive, the color scheme doesn't make a huge amount of sense. But it is showing you the different magnitudes of fold change. So that worked quite nicely, actually. Just to get that extra bit of data into that plot. I'm not saying that the layout of this plot is optimal, because I really don't think it is. But that for just a kind of one-liner based on this function, which the cluster profiler package gives us for visualizing that data, that's an okay plot. Let's have a look at this Go plot as well, because we haven't seen this before, or I haven't shown you this before because I couldn't get it to work the first time around. So hopefully it's going to work this time. So this is representing the gene ontology, and the gene ontology that is represented in our enriched categories. So we might need to talk a little bit about the gene ontology to understand this plot. Let me make it bigger, see if it lays out a bit better. So the gene ontology is what's called a directed acyclic graph, which is otherwise known as a hierarchy. So directed means that the edges in the gene ontology have a direction, so you go from one to the other, and you generally proceed down through a hierarchy. So multi-organism process here is a child of biological process. So the relation here is biological process has child, multi-organism process has child, interspecies interaction between organisms, etc. as you work your way down this tree. So the further you work down, the more specific the gene ontology terms become. And then this is showing you the enriched categories in this particular analysis, coloured in according to the p-value of that enrichment. So you've got the more red the p-value is, the more enriched that particular term is, effectively, in this kind of blue through purple to red colour scheme. You can see that, and this is generally true, and this is true of these kind of enrichment calculations anyway, that there's a lot of relationships between the gene ontology categories which are enriched in this particular analysis. And that's because, generally speaking, the children term are effectively a subset of the parent. So all of the genes that are represented by the regulation of biological process term are also represented in this negative regulation of biological process term, and so on as you work your way down. It's like a funneling effect. Your categories get smaller and smaller as you go down. But if you're overrepresenting this term down at the bottom, there's a greater chance that you are overrepresenting its parent and its parent and its parent, because A is a subset of B, is a subset of C, is a subset of D. So the fact is that each of these terms contains the same set of genes plus a few more or a few less, depending on where you are in the tree. And so as you proceed up this tree, the chance that you'll be overrepresenting a particular term gets less because you're introducing more noise into here. But once you've found overrepresentation at one level, then the chances are that a lot of those children are also going to exhibit overrepresentation as well. So you end up getting these kind of nests of overrepresented terms in the gene ontology graph. But this can be quite a useful way of visualizing that, because you can see those neighborhoods of overrepresentation and also how they're related to one another. So this is quite a useful way of visualizing that data. Okay, so what I would like to do before we finish is to begin to think about how we compare these two experiments. So we have now, apart from the KEGG analysis, which I'm not going to repeat right now, because I feel like we have repeated a lot of things and it is doing the same things over again. And so to spend 3 days on effectively repeating a whole load of stuff we've already done, I realise, and I notice that the audience is lower today. Hello to those of you that are here. So at this point, I'm going to draw a line under that repetition. And what I'd like to do therefore, is start to think about how we compare analysis A with analysis B. Okay, and so we're going to start a third script, and I will upload this second script to GitHub this evening once we've finished here, to make sure that everybody can get up to speed. We're going to start a third script which depends on the first two scripts. Okay, so it's going to rely on those first two scripts having been run, but then starts to pull out some comparisons between analysis A and analysis B. Okay, so let's close this and let's start another new script, and then start to think about how we're going to run this. So I'm starting this from scratch, so I'm not going to copy across anything to this particular script. But as I said before, so not even the package declarations at the top of the script. I know we are going to need the tidyverse, so maybe we can load that. But other than that, I'm going to leave it until we actually know that we'd need a particular package. Okay, so we are going to need the tidyverse because we're going to do some plotting. And that's the first thing that we're going to do right now, is we're going to produce a plot of a comparison plot between these two analyses. So we're going to load the tidyverse. But then this script is going to depend on the other two scripts in this analysis. So those two scripts are going to have been run before we can do anything in this script. And you can do that from within R. You can run another script from within your current script using the R function called source. So source will run the script which is its first argument. Okay, so if we source COVID RNA analysis.r, what this will do, what that line will do, is it will run in its entirety that COVID RNA-Seq RNA analysis script, and all of the variables that are created when that script runs will all be loaded into our environment. So we can be sure that that script has run and that all of the variables that are created while it's running are then available to us within this script once we want to start using those variables. And we also want to run the second analysis as well. So we also want to run, this needs to depend on the COVID A549 analysis as well. And we need all of the variables from that analysis. Now we know for that one we've already got those in our environment because we've only just finished writing that script and we've got all of the results of that in our environment already. But if we came back to this script in 6 months' time when we've done other things, we couldn't depend on that. So we're kind of setting up a dependency here between this script that we're going to start writing now and the two scripts that we've written before, the two scripts that we've already generated previously. So this is a relatively useful thing to do and can stop your scripts getting absolutely monolithic. So it can be the case that over the days and weeks and months potentially of doing an analysis like this, which is very exploratory in nature, and you try things and you plot things and then you go back and you try more things, and lots of that ends up in your script. That without going back and really tidying it up, you can end up with these huge monolithic scripts of thousands of lines of R, which can be really, really hard to navigate. And so breaking your scripts down into these individual parts, which depend on one another, can be quite a useful way of kind of dividing your analysis up a little bit. So let's save this script before we go any further and give it a name. So this is going to be COVID comparison. So just move my save dialog so you can actually see it. Comparison analysis. Okay, so let's save that so we've got that copied as well. Okay, so we've run the second of these two analyses already because that's what we've been doing over the last few days. But I cleared my environment before I started running that, so I don't have the equivalent data from this first analysis. But so I'm gonna run that source there, and hopefully what you can see in my console here is that it's working its way through that script. It's not telling me what it's doing, so it's not printing out the lines of code as it's running them, but it is printing out the results, and it will be loading those variables that are created as that script runs into my environment. So once we get through to the end of running this, we will find that we've got all of those NHBE variables in the environment, as well as all of the A549 variables in the environment as well. So this is just running through that code now. And has that got to the end? Or doesn't look like it's got to the end. What's happened there? Okay, let's just see if we can do... If you set an argument for echo to true, then it will print every line of the analysis. It'll print the code as well as printing the results as it goes. And that might be quite useful to track down what went wrong there, because something failed. And it may be, this was quite a... This script had no tidying up at all. We didn't tidy it in the slightest as we went. So it may well be that that lack of tidying has resulted in some sort of failure at some point that we haven't caught. So this time it's running through and it's printing all of the code as it goes. It actually seems to be working better, potentially. Oh no, there you go, it's failed. Why has it failed? It's failed after the k-means clustering. Let's have a look at the script. K-means clustering. One of the few things we didn't actually repeat. DDS1 estimate dispersions. That we knew failed. We know that failed because it says underneath it, the above doesn't work. So we need to comment that out, or maybe delete it, but at least comment it out. And And that's part of the problem of this kind of iterative development, where you're kind of developing as you go. If you forget and leave these lines in that don't actually work correctly, then this can really cause knock-on effects. Off we go again, then try again. What I want is to get to a point where we have it's the first data frame, the first filter in your gene expression results, where you've got all of the genes for which you've calculated a log fold change in DESeq2. So all of the genes that are expressed that have passed all your filters in DESeq2. I want those two sets for the two data sets to be able to compare the fold change in experiment 1 with the fold change in experiment 2. Because that's kind of our first test for concordance between these experiments, is although we may not find the same set of differentially expressed genes, we would expect to see that the trends in differential expression are relatively similar between the two data sets, that things that are trending up in one data set are also trending up in the other data set. So I want to plot the fold change in the NHBE against the fold change in the A549 cells, and see how that looks, see what that looks like. So we've got to put that together, that data set together, to form that analysis. So you can see that this is running, and we're just getting to the end now. So this is doing the enrichment. So that's at the very end. So now we should have our environment. We should now have all of the data that we need to start producing this plot. So I'm going to clear my viewer here because that slows everything down. Okay, here we go. So if we look at our environment now, we should find that as well as all of our A459 objects, have also our NHBE objects in there as well. So all of our variables from both of our runs through are present in our environment now. So hopefully that includes our first filter results for both the A549 experiment, for which we have about 15,000 results, and our NHBE experiment, for which we have a similar number. It's actually about 16,000 results for the NHBE experiment. And what we want to do there then is compare. So we've got annotated DF. This is the NHBE first filter results. And we've got the A549 first filter results. That is the excuse me, so A549 ANODF. Okay, that's that one. A549 ANODF. Okay, so those are our two data sets that we want to pull the data out of in order to generate this plot. But we can only really do that where they share data, where the same genes are represented in both data sets. So where we've got genes that are missing in one data set versus the other, then oh, that one I can answer. Sorry, I've seen a few questions go by as we've gone. You need to load the that package. So you need to do library, Manuel, you need to do library org.hs.eg.db to load that annotation package into your R session. So this line here is what you need to run to make that package available in your R session. So that may be missing at the top of your at the top of this script. If that's you rerunning this script, then that library statement may be missing at the beginning of that script. Okay, so sorry, diverted there. We can only really do this for genes which appear in both datasets. We want genes which are in both the NHBE dataset and the A549 dataset, because if they're not together in those two datasets, then we'll have a value in one and an NA in the other, and the plotting won't work very nicely because of those NAs. So what we want then is the ensemble gene IDs that are shared between these two datasets. So let's begin by getting those ensemble gene IDs. So we want NHBE ENS gene is the annotated DF, and it's the ENS gene column of that data frame. Okay, so that's the genes that are in that data set. And then we want the same, so A549 ENS gene is from A549 anno DF. We want again the ENS gene column. So that gives us two vectors of ensemble gene IDs of different lengths and with different contents. Okay, so the set of these genes, they should overlap considerably, but they won't be exactly the same because we already know that they're different lengths. And we can find the ones in common using set arithmetic. So we can treat the NHBE ENS gene vector as a set, and the same for this second vector as well. And we can ask, using set arithmetic in R, we can ask what the intersection between them is. So intersect is the way of doing this. And it's the intersect of these two. It's like if you drew a Venn diagram, the intersect is the region in the middle of the Venn diagram, the overlap between the two sets. So that the intersect of those two vectors, which looks like it's about 13,500 ensemble gene IDs, that is the genes in common between our two analyses. So this is common ENS gene. Okay, so what we now want to do is, if we want to plot the fold change of one against the fold change of the other, then we want to create a data frame that contains the fold change from the NHBEs and the fold change from the A549s for these common genes. Okay, for these 13,500 or so common genes. The way that we get that, therefore, is we need to filter our data frames. So we want our annotated data frame. We want the rows, so before the comma, we want the rows where the annotated DF, the ensemble gene ID is in. Have we done this before? So the ensemble gene ID is in this set of common genes. So every entry in here, which is also contained somewhere in here, this statement returns true. and for everything else it returns false. So this will give me 13,500 rows of this annotated DF. And then the what we want in the columns then is the log to fold change. So we also want to filter the columns in this case. So we do want something after the comma, and in this case we can just use a column name. So this will give me a vector of fold changes for those genes. We need to make sure these are in the same order, don't we? Okay, let's get the ensemble gene IDs as well. Okay, hold on, let me show you this, because we need to make sure they're in the same order. Okay, so that gives me, sorry, a 2-column data frame. Why did that not work? Because it all needs to be on the same line in this case. No. Unexpected. Oh, didn't close the bracket. There you go. So that gives me a 2-column data frame of ensemble gene ID in one column and the fold change in the second column. We want to do the same, so that's our NHBE common genes. And then we want to do the same for the A549. So the A549 common genes is the same, effectively the same thing, and we want to get it from our A549 anode DF. Sorry, I may have bitten off slightly more than I can chew in the time that I had. I do want to get through this, so I might speed through this and then re-row, if that's okay with everybody. Right, okay, so that's our A549 common genes. That's our NHBE common genes. And then we can use join to link two things up. So we can use a left join for the NHBE common and the A549, where ends gene equals ends gene. I just want to be explicit about that, because just in case any two genes for some bizarre reason share a log fold change, in which case the join gets much more complicated. But that will give me my 13,000 rows with, if we have a look at it, it will have the ensemble gene ID in the first column, then the fold change for one, for X, which is the left-hand side, so NHBE common in one column, and the fold change for the other in the next column. So this is my common fold changes. And then that's what I want to plot. So I want to ggplot. This is my last thing today. So the data is common fold changes. The aesthetics, the x-axis, is the NHBE fold change. So log2 fold change dot x, and the y is the log2 fold change dot y. That's the A549 fold change, and then we want to add a point geometry. So we're going to produce a scatter plot, and I'm just going to make, because this is going to be over plotted, I'm just going to decrease the alpha so that we can see a little bit of overlapping. Okay, so I realise I've raced through this a little bit, but I think I bit off slightly more than I could chew this afternoon. We actually have to run the left join. Let's make our plots window bigger, because this is going to be quite daunting. And off we go. So this is going to look at a little bit at the concordance between our two data sets. And face value, that doesn't look too great. But actually, it's probably slightly misleading because of the outliers pulling it in various directions. So we'll work with this tomorrow then and see if we can figure out if these are actually correlated or not, whether these fold changes are correlated or not. So yeah, you've got so the most changed gene in the NHBE cells has basically 0 fold change in the A45549 cells, and that's really pulling this really skewing the axes here. And actually the same is true for the A549 as well. The most changed genes there are not really changed at all in the NHBE samples, although, well, maybe a couple of fold. So a little bit of work to do with this plot, but this is a starting point, okay? So we'll work with this and try to make it a little bit more informative tomorrow, and try and figure out if the fold changes between these two experiments is even slightly correlated. Okay, I'll leave it at that today. Sorry I've gone on a bit. I didn't mean to go on for quite so long, but I think I got slightly ambitious towards the end. We'll work more on this plot tomorrow, and we'll do some other comparisons as well. Thanks for tuning in today, and I hope to see you again back again tomorrow.